Thank you for the introduction. Hello, my name is Lei Zan. I'm the Deputy Director of the Office of Research and Standards in the Office of Generic Drugs at the FDA. Today, my colleague, Dr. Nilufer Temple, Associate Director of Scientific Quality from the Office of Bioequivalence in the Office of Generic Drugs at the FDA, and I will present to you this webinar in collaboration with the SBIA on the recent FDA revised draft guidance on bioequivalence studies with pharmacokinetic endpoints for drugs submitted under an ANDA. The vast majority of bioequivalence studies submitted under an abbreviated new drug application to the FDA are pharmacokinetic studies for which this guidance is the key resource. Here is the outline of today's webinar. We will provide you with the objectives of the webinar, a guidance history and scope, overview of the guidance, a summary of major changes in this revised guidance. We will also provide you the key topic areas with questions and comments received through the docket. We will also provide responses to selected questions and comments we received. We will end the presentation with a summary. After the presentation, there will be a panel discussion with additional panelists from the FDA, and we will also have a question and answer session to answer the audience questions. Here are the objectives of today's webinar. We will provide an overview of the revised FDA draft guidance for industry titled Bioequivalence Studies with Pharmacokinetic Endpoints for Drugs Submitted under an ANDA that was published in August 2021. We will describe the major changes in the revised draft guidance. Additionally, we will provide clarification and the rationale on selected topics to address comments we received through the docket. Finally, we will help the audience understand how principles described in the revised draft guidance in conjunction with product-specific guidances and other pre-submission communications can facilitate generic drug development and generic drug application assessment. We do not intend to cover every aspect of the guidance, especially those parts that have not been revised. We do reserve some additional time through the panel discussion to provide additional clarifications as needed. Bioavailability or BA assessment of drug products is a component of new drug development. Bioequivalence or BE assessment, on the other hand, is important for establishing therapeutic equivalence for generic drug products to their respective reference-listed drugs. In addition, application of the BE concept is important for bridging various formulations during drug development. Furthermore, BE studies are used by innovator and generic drug product developers to bridge certain types of post-approval formulation changes. The BE concept is core to product development and regulatory assessment for generic and new drug products throughout their life cycle. The FDA's 2003 final guidance titled Bioavailability and Bioequivalence Studies for Orally Administered Drug Products General Considerations covers both BA and BE and was applicable to both NDA and ANDA applications. Since this guidance was issued, FDA has determined that providing information on BA and BE studies in separate guidance according to the application types would allow FDA to provide more specific recommendations pertinent to the application types, which would be beneficial to sponsors and applicants as well. In 2013 and 2014, FDA issued two separate draft guidances with one focus on the new drugs showing on the left and one focus on generic drugs showing on the right with different guidance titles. Both these two guidances have gone through revisions since 2013 and 2014 respectively. The most recent revised draft guidance for new drugs was issued in February 2019 with a revised guidance titled Bioavailability Studies Submitted in NDAs or INDs, General Consideration. FDA recommends that applicants for INDs, NDAs, or NDA supplements consult this guidance. The most recent revised draft guidance for generic drugs or ANDA was issued in August of last year, which is the topic of today's webinar. For abbreviation, we will call this guidance Draft ANDA Pkbe Guidance. The Draft ANDA Pkbe Guidance provides recommendations to applicants planning to include BE information in their ANDAs and ANDA supplement submissions. It describes how to meet the BE requirements set forth in the Federal Food, Drug, and Cosmetic Act and FDA regulations. It's generally applicable to dosage forms intended for oral administration and also to non-orally administered drug products in which reliance on systemic exposure measures is suitable for establishing BE. For example, transdermal delivery systems and certain rectal and nasal drug products. It also covers BE studies conducted during post-approval period for ANDAs. Although BE concepts could be applied for new drug development, as I mentioned earlier, FDA recommends that applicants for INDs, NDAs, and new drug application supplements consult the other draft guidance for industry titled Bioavailability Studies Submitted in NDAs or INDs, General Considerations, which addresses bioavailability studies for these submission types. This would include 505B1 and 505B2 applications. For generic drug applications, applicants can consult the product-specific guidances or PSGs as another resource. PSGs reflect the FDA's current thinking and expectations on how to develop a generic drug product therapeutically equivalent to a specific reference listed drug, ILD. FDA recommends ANDA applicants consult routinely published PSGs when considering the appropriate BE studies and or other studies for a proposed drug product. They can refer to the FDA's product-specific guidance webpage, as shown here, for the most recent version of our PSGs. Recently, FDA also held an SBIA webinar on the PSGs, which can serve as a good reference. Product-specific guidances align and reflect both the FDA regulations and general guidances. FDA's regulations, general guidances, and PSGs altogether provide current BE approach for a specific product. Here is a high-level comparison between the 2013 draft and the 2021 draft and the Pkbe guidance on a high level. By looking at the section from 1 through 3, there's no change. For section 3B, the only change is that the order of the subsections by moving the in vitro studies up front. The change in section 4, establishing bioequivalence for different dosage forms, include addition of 3 subsections, which is orally disintegrating tablets, sublingual, and transdermal. The change in section 5 includes removal of the subsection orally administered drugs intended for local action and addition of a new section entitled Enteral feeding tube. Finally, there's changes in the Appendices section by adding 2 new appendix sessions. Appendix B, talking about method for statistical analysis using the reference scaled average BE for highly variable drugs, and section Appendix C, talking about method for statistical analysis using the reference scaled average BE for narrow therapy index drugs. In the next few slides, I will provide you with an overview of the ENDA Pkbe Guidance. First of all, it states about the general BE principles, which is the rate and extent of absorption of the drug do not show a significant difference from the rate and extent of absorption of the listed drug when administered at the same molar dose of the therapeutic ingredient under similar experiment conditions in either a single dose or multiple doses. The guidance also talks about both in vivo and in vitro methods can be used to establish BE, which include comparative PK endpoint BE studies, in vitro tests that are predictive of human in vivo BA, and comparative pharmacodynamic endpoint BE studies, comparative clinical endpoint BE studies, and also in vitro studies. The guidance also covers the elements on the BE study design, which is mainly covered under Section 3 of the guidance. In addition, in the Appendix A, there are also description on the general design and the data handling of the BE studies with PK endpoints. That covers the study conditions, which including fasting versus fed conditions, sample retention, sample collection, and sampling times, and also in which biological matrix. What PK information should be included in ENDA submissions, and also the data analysis elements. In terms of study design, the guidance covers what are the appropriate study populations, healthy versus patients, and what's the consideration in the study population with regard to age and sex. And there are some revision with regard to age and sex that will be discussed more later. In terms of study types, the guidance talk about when we should consider single dose or multiple dose, crossover design versus parallel study design, non-replicate versus replicate study design. In general, the single dose is considered more sensitive to detect the formulation difference. And we recommend a 2-period, 2-sequence, 2-treatment, single-dose crossover study. For drugs with long half-life, a single-dose parallel studies design may be appropriate. And we also discussed what is a replica design for BE studies is most applicable, which I will describe in the next slide. The guidance also covers the recommendation on the sample size considerations and study conditions with regard to food, what dose strength to be studied, and also safety considerations, which is also relevant to the consideration on whether to enroll healthy subjects or patients for a specific product. The revised draft guidance provides more clarifications with regard to replicate study design. It clarifies that a replicate crossover study design, either partial replicate or fully replicate, is appropriate for drugs whether the reference product is a highly variable drug or not. A replicate design can have the advantage of using fewer subjects compared to a non-replicate design. However, each subject in such study would receive more treatments. The guidance recommends that a replicate design is to be used under the following 2 scenarios. The first scenario is when the reference product is a highly variable drug. the design can be either partial or fully replicate. And the reference scaled average BE biostatistical analysis approach can be used, and more details is provided in Appendix B. The second scenario is when the reference product is a narrow therapeutic index drug. The guidance recommends a fully replicated study and with the reference scaled average BE. More details is provided in Appendix C with regard to biostatistical analysis. With such fully replicated study design, it will be possible to compare the inter-subject variability of the test product and the reference product. BE studies conducted under the fasting conditions can help determine the potential formulation impact on the absorption or bioavailability of the test and the reference products. Food can impact oral absorption and may impact oral absorption differently depending on the product formulations. Therefore, BE studies conducted under fed conditions can help determine whether test and reference products are bioequivalent when they are co-administered with food. For an orally administered immediate release product, the guidance recommends that applicants conduct both fasting and fed BE studies, with the following exceptions. One is when the RLD labeling states that the product should be taken on an empty stomach, or when serious adverse events are anticipated under the fed conditions. In these situations, a fed BE study is not recommended. Another exception is that when serious adverse events are anticipated with fasting administration. FDA recommends that applicants conduct only a fed BE study in this situation. A fasting BE study is not recommended. Advancement in the science and research to understand interactions of food and formulations can help determine proper BE testing methods. Currently, recommendations related to fasting versus fed BE studies are under discussion as part of the harmonization efforts under the International Council for Harmonization ICH M13 Expert Working Group. For modified released oral drug products, the guidance recommends that applicants conduct both fasting and fed BE studies irrespective of dosing instructions in the ILD labeling, with the following exceptions. One is that when serious adverse events are anticipated under fed conditions, a fed BE study is not recommended. In such situation, BE is recommended to be conducted under fasting conditions only. The second exception is that when serious adverse events are anticipated with fasting administration, the guidance recommends that applicants conduct only a fed BE study. In this situation, a fasting BE study is not recommended. As I mentioned earlier, this guidance covers BE considerations for post-approval changes. FDA has separate SUPAC IR or SUPAC MI guidances to dictate the level of changes that may warrant in vitro or in vivo BE testing. For post-approval changes in general, FDA recommends that applicants make the in vitro comparison between the pre-change and the post-change ANDA products. When in vivo BE studies are recommended based on the level of changes to support a post-approval change, FDA recommends that applicants compare the post-change ANDA product to the reference list drug and not to the pre-change ANDA product, because the reference list drug is the reference standard to be used for in vivo BE testing. Now I will turn over to Niloufar, who will present on the summary of major changes in the August 2021 draft and the Pkbe Guidance.
Thank you, Lei. Hello, everyone. My name is Nilofer Tempal. I'm the Associate Director for Scientific Quality in the Office of Bioequivalence. We all heard Dr. Zhang describe the scope and the background for the revised guidance and give a nice overview. Now we are going to shift gear to give a summary of the major changes in this guidance. And while I will briefly provide clarifications and rationale for some of the changes, we plan to get into more details for these during the panel discussion. So I'll start with the first two sections, which are the introduction and background sections of the guidance. In these sections, the guidance updates and clarifies the content on the reference-listed drug RLD and the reference standard RS identified in the Orange Book. And it also references new guidances, such as the 2019 draft guidance on bioavailability studies in NDAs and INDs, and the product-specific guidances, the PSGs. The PSGs and the general BE guidance are closely related. FDA routinely publishes the PSGs, and we recommend that in addition to the general BE guidance, ANDA applicants consult the PSGs for the appropriate bioequivalence studies for their proposed product during the drug development. Let's look at the updates for the RLD and the RS. For BE studies, an applicant generally will compare the systemic exposure profile of the test product to that of the RLD, which is designated in FDA's approved drug products with therapeutic evaluations. We commonly refer to this as the Orange Book. And then RS is the specific drug product selected by the FDA that the applicant must use in conducting in vivo BE testing to support their approval. Typically, the RS selected by the FDA is the RLD. However, when the RLD is withdrawn for reasons other than safety and efficacy, and therefore not available on the market, the FDA may designate an approved generic product as the RS, which the applicants must then use in the study. If you want to further understand the distinction between the RLD and the RS, you may refer to the guidance on referencing approved drug products in ANDA submissions. Next, in Section 3 on establishing bioequivalence, the guidance expands the content on study population with regards to sex and age. With regards to sex, the guidance mentions that if a drug product is intended for use in both sexes, the applicants should include similar proportions of males and females in the study, or provide a justification supporting the use of a single-sex population. Now, this recommendation for similar proportions of males and females is not a new recommendation, and no new science has been introduced here. This recommendation shows up in our 2013 guidance, and it goes as far back as the 2003 guidance. What's new here is the justification part. So why was this recommendation introduced? Well, FDA is increasingly observing that studies done in certain parts of the world tend to use a single-sex population, while the drug is meant for a broader population. As a scientific community, we all acknowledge that the results from a crossover BE study are generally transportable. However, when there is a deviation from the recommendation, for example, not using males and females in the study, then the guidance recommends that the applicants provide a justification to support how your conclusion for the bioequivalence of your test product in one group correlates or is transportable to the bioequivalence in the other group. And I suggest to hold your thoughts on the justification part for now, and you will hear more on FDA's thinking on this from the panelists during our panel discussion. So continuing, the guidance further mentions that if a drug product is intended for use in a single sex, then the applicant should only include subjects from that sex. And for safety reasons, females should not be pregnant or lactating, and if applicable, practice abstention and contraception. Now, with regards to age, the guidance includes recommendations for both the elderly and the pediatrics. So if a drug product is predominantly intended for use in the elderly, then the applicants should include as many subjects as possible at or above the age of 60, and if that's not done, then they need to provide a justification. Again, the recommendation for including the elderly also appears in our 2013 guidance. The major update here is the language for including the justification, and the rationale for this additional language for justification is similar to what I mentioned earlier for the single-sex population. Coming to the pediatrics. For pediatric in general, the bioequivalence assessment in adults can be used to support the bioequivalence assessment in pediatric patients. Here, the guidance recommends that if the drug is predominantly used in pediatrics younger than 6 years, the applicant should justify that the results from the adult subjects are relevant to the pediatric population. And FDA also recommends that this justification include information to support the inactive ingredients in the test product are appropriate and safe for use in the pediatric population. Now moving on, under Section 4, the guidance modifies the recommendations for assessing proportional similarity for additional dose strengths for modified release drug products based on considerations of formulation, composition proportionality, and the mechanism of drug release. So here we have the schematic showing the recommendations in the 2013 draft guidance and the 2021 revised draft. In the 2013 guidance, demonstration of bioequivalence for additional dose strengths for MR was based on showing proportional similarity, acceptable dissolution, and the same release mechanism. And proportional similarity in formulations was defined very similarly for the IR and the MR products. Now, in the current guidance, this language is modified such that the differences in the composition proportionality of the formulations between additional strengths and the biostrength of the MR product may be justified now, and they may be shown to be appropriate based on the drug release mechanism. Now this slide shows the different considerations for determining bioequivalence of additional strengths for the MR products. So firstly, the bioequivalence should be demonstrated based on in vivo studies for the biostrength. And then we take a number of additional factors into consideration when determining the bioequivalence of additional strengths, right? So here, first to start with, for the reference drug product, we look at, does the RLD exhibit consistent bioavailability in terms of the dosage form equivalence, and does it show similar dissolution across all strengths? For the test product, we want to see, are the additional strengths formulated to have the same drug release mechanism as the biostrength? Also, are the dissolution profiles similar between the biostrength and the additional strengths at least 3 different pH media? And then, are the excipients qualitatively the same across the strengths? And also, are the ratios of the drug to the excipient among different strengths can they be justified per the drug release mechanism? So formulation composition proportionality may not be the only factor to determine the need for an in vivo bioequivalence study to demonstrate the bioequivalence of additional strengths. We would take the totality of the evidence into consideration, which would include the bioequivalence on the biostrength, formulation evaluation, and dissolution data in order to conclude the BE, if BE of the additional strength has been demonstrated. So this is the language directly from the guidance, which talks about the data that may be needed to support bioequivalence of additional strengths. And I have put in bold the key points here. The first is for the reference product that we need to see the dosage form equivalence and among the different strengths and similar dissolution profiles. For the test product, the excipients should be qualitatively similar among the different strengths. And as we said earlier, the ratio of the drug to the excipient of different strength can either be proportional or it may not be proportional, but can be justified and may be appropriate based on the drug release mechanism of the test product. Also, the additional strength should have the same drug release mechanism as the biostrength, and then we want to see we would check the similarity in dissolution profiles in at least 3 dissolution media for the test product. And although we look at the dissolution similarity across the strengths of the test product line, the guidance recommends that applicants generate dissolution profiles on the test and the reference products. And determination of whether the totality of evidence supports BE will be made during the ENDA assessment. And there may be instances in which the in vivo BE study for non-proportionally formulated strengths may be necessary to demonstrate bioequivalence. The next major changes are all new additions to the guidance. And although they are new for the 2021 guidance, they are not really new in the sense that they have been in practice for a while, and they are also mentioned in different PSGs. They were just missing from the 2013 guidance, and now we have included them in this guidance for completeness. So first, under section 4, new dosage forms that also rely on systemic bioequivalence assessment have been added. And these include both the oral dosage form, like the ODTs and the sublinguals, and the non-oral dosage forms, like the transdermal drug delivery systems, for which we rely on systemic exposures to establish bioequivalence. For oral ODTs and sublinguals, similar to the chewable tablets, the guidance recommends how the applicants should administer these products in the in vivo BE studies. The next addition is for the enteral feeding tube, and it's under section 5. The guidance introduces recommendations for the in vitro test for enteral feeding tube, which includes the nasogastric, the NG tube, and the gastric, which is the G tube. Again, these are not new recommendations. Many of the PSGs include details of the in vitro test for this alternate route of administration. We also now have a general guidance on this topic, and this was published around the same time as the general BE guidance. And this guidance, titled Oral Drug Products Administered via Enteral Feeding Tube, includes the recommendations on the in vitro testing and labeling, and you may refer to it for further details. Now coming to the appendices, Appendix A includes a new section on handling of the outliers. And the guidance states that the applicant should not remove data from BE analysis solely based on the fact that the data is identified as a statistical outlier. Outliers may only be removed if there is a real-time documentation demonstrating a protocol violation either during the clinical and/or the analytical phase of the BE studies. Also, applicants should include a prospective plan in the study protocol for removing the outliers. Redosing studies are not considered as evidence to support the removal of outlier data. And finally, all subject data should be submitted, including the potential outlier data, and these should be flagged with appropriate documentation in the submission. Also, 2 new appendices have been added to the guidance. Appendix B describes the method for statistical analysis using the reference scale average BE approach for highly variable drugs, and Appendix C does the same for the narrow therapeutic index drugs. Previously, these methods were included in the PSGs for progesterone oral capsules and warfarin sodium oral tablets, respectively. However, going forward, the future PSGs may reference the appendices in this general guidance for this purpose. Lastly, since this guidance is intended to focus on Pkbe studies for drugs that are systemically acting, as mentioned earlier, the section on the orally administered drugs intended for local action, which was in the 2013 guidance, is now not included in the current 2021 draft guidance. And that's because for some locally acting drugs, comparative clinical endpoint or pharmacodynamic endpoint or even in vitro tests may be appropriate to demonstrate bioequivalence. At the same time, there may be some cases, such as the locally acting GI drugs, where determining bioequivalence using the PK endpoint may be relevant. In such cases, the principles in this guidance would still be applicable. And in future, FDA may develop a separate guidance on the locally acting drug products. Now, this slide summarizes the major changes that I covered, and it was put together as a ready reference if needed. With this, I will turn it over to Dr. Zhang, who will now walk us through some of the comments and the questions we have received on this guidance through the docket. Lei, back to you.
Thank you, Niloufar. So FDA received approximately 20 comments through the docket. Below are examples of the key topic areas with comments and questions. We have received comments on expanding the scope of the guidance to include complex formulations like inhalation products, liposomal products, and vaginal cream. Comments and questions related to study population, BE studies with regard to food, Tmax comparison, additional strength waiver for the IR product when active and inactive ingredients are not in similar proportion, and BE demonstration for additional strength for the MR products, and question about alcohol dose dumping. Elaborate more about when an Nvivo study may be needed, especially when there's no PSG, and a comment regards to guidance on sample retention. So we will cover some response to the comments questions in the next few slides, but we also will address some of them during the panel discussion, so we can have more elaboration on what should be considered. Here is the response to the first comments relate to expand the scope of the guidance to include complex formulations like inhalation products, liposomal products, and vaginal creams. So this guidance focuses on PK endpoint BE studies. The principle can be used to guide complex products as mentioned above if the PK endpoint BE study is being used for BE demonstration. However, for these complex products, they will need additional data to support BE, especially if they are locally acting. Currently, the PSG may be the best source to understand FDA's current thinking and expectations on how to develop a generic drug product therapeutic equivalent to their respective RLDs. So we would recommend applicant to refer to specific product-specific guidance. And also for complex product, if there's no PSG, the applicant can request pre-ENDA meetings to meet with the FDA for recommendations to their drug development program. In this question, it asks FDA whether FDA can provide further insights as to how FDA is defining non-smoking. Does non-smoking mean a subject that has never smoked, or does it also include a subject that was a previous smoker or has had history of smoking but has stopped for a duration of time prior to the study? So in general, subjects who have never smoked are preferred for the BE study. However, subjects who have had a history of smoking but have stopped for a duration of time, for example, at least a year prior to the study, may be acceptable, especially for drugs that are not substrates of enzyme that are induced by smoking, for example, CYP1A1, CYP1A2, or CYP2E1. So if this drug is not a substrate of these enzymes, then their PK is less likely to be impacted by the smoking status of the subject. In this question, it asked, is a statistical comparison required for Tmax, or is the magnitude of difference a subjective evaluation? So the answer is that no, the statistical comparison is not required for Tmax, and the Tmax assessment is not subjective. FDA takes several approaches to evaluate the impact of significance of Tmax differences of the test and reference products, including clinical consideration of the product. In some cases, if the rapid onset is important, partial AUC may be calculated and subject to statistical comparison to assist the decision-making. The last comment received I would like to cover in my presentation is related to retention of BA and BE testing samples. The comment asks FDA to reference a more recent guidance for industry titled Compliance Policy for the Quantity of Bioavailability and Bioequivalence Samples Retained under 21 CFR 320.38c issued in August 2020. This comment is noted. The new guidance mentioned above that is published in August 2020 is to clarify the number of test and reference products to be retained under 21 CFR 320.38. This PK guidance does not touch on that aspect. Here, the Pkbe Guidance is to specify the length of the time the sample needed to be maintained per 21 CFR 320.63. So the proper guidance is cited related to sample retention pertaining to this Pkbe Guidance. In summary, the Pkbe Guidance is the key resource for ENDA applicants where Pkbe studies are used to support BE. ENDA applicants should refer to general guidances or PSGs for BE approaches for a specific product. They can submit questions via controlled correspondences if need FDA's input on their drug development program. In addition to controlled correspondences, FDA provides multiple communication channels, including product development meetings pre-submission meetings, mid-cycle review meetings, and post-complete response letter meetings at various stages to address industry's questions to help generic drug development and assist regulatory approval. Here are a few resources that you may find helpful, including the link to the FDA guidance webpage and a few guidances that are relevant to the PK and the BE guidance. Here are the resources relevant to the product-specific guidances, including the link to the PSG website and other relevant information. This concludes our presentations. Thank you for your attention. We will be happy to answer your questions, followed by a panel discussion. Thank you.
Thank you so much, Dr. Zhang and Dr. Temple, for the excellent and informative presentations. My name is Lucy Fang, and I am the Deputy Director of Division of Quantitative Methods and Modeling, Office of Research and Standards. I will moderate the panel discussion session today. In addition to the two speakers, we also have other OGD colleagues joining the panel session. Dr. David Coppersmith, who is a Regulatory Counselor from the Office of Generic Drug Policy. Dr. M.J. Kim, who is a Division Director from the Division of Therapeutic Performance 2, ORS. Dr. Bing Li, who is an Associate Director for Science from the Office of Bioequivalence. Dr. Robert Leinberger, who is a Director from the Office of Research and Standards. Dr. Diana Vivin, who is the Acting Associate Director from the Office of Bioequivalence. Dr. Niang Zhao, who is the Division Director from the Division of Quantitative Methods and Modeling, ORS. Please join me welcoming the panel members. Let's come to our first panel question. Can you comment on how do FDA develop product-specific guidance, PSG, in alignment with the general guidance? What if there are differences in recommendations between PSG and the general guidance? Dr. M.J. Kim, can we come to you first?
Thank you, Lucy. Hello, everybody. This is MJ Kim. I am mainly working on product-specific guidance development. So I am very excited to be here to share with you in terms of how we develop PSG guidances in alignment with the general guidance. So as you can tell, the general guidance is somewhat of a kind of big umbrella as to how the document conveys the overall thinking and key principles for establishing bioequivalence. But for product-specific guidances, and you should not be surprising news to you, but each product has its uniqueness. So when we develop one PSG at a time, we utilize the general BE guidance, the underlying key principles, and then we look at the studies submitted under the new drug applications, and then the control correspondence that we receive and the previous ones that we have responded. And then we also look at other product-specific guidances of same drug products with the drug product with the same API, or the drugs in the same class of drugs. And of course, most importantly, we look at the drug labelings as well. So utilizing all that information, we develop and we come up with specific recommendations in terms of study populations and what are the specific PK parameters that we should consider if it's not really captured in the general guidance. And you have seen in the slide deck that we have a specific topics such as ODT, chewable, and other products. And then and when we develop PSGs, we try to give a very specific instruction as to how to do a BE studies. And there were some questions about the guidances from the other regulatory bodies. As one can imagine, we do look at those guidances documents as well, and we try to understand the rationale and the recommendations from that as well. But once again, each product-specific guidance is supposed to be dedicated for the one product at a time, and each product has its uniqueness. So we try to be very specific and provide useful information for the generic drug developers. With that, I'll stop here. Thank you, Lucy.
Thank you, Dr. Kim. Dr. Robert Lineberger, you have any additional thoughts in this regard?
Hi, this is Rob Lineberger. Just to re-summarize that you have, the audience here should really understand that the general guidance and the product-specific guidances work together. There's not one that's higher or lower than the other. They work together, right? In most cases, the product-specific guidance follow the general guidance, but provide specific details on implementation, like which metabolite to measure, exactly, how to dose with, like as MJ gave the example, how to dose with water. In other cases, the product-specific guidance, if there is something that's unique about that product that's different than the general approach, then you'll also find that in the product-specific guidance. So they work in alignment, and we put significant effort in keeping them in alignment and keeping the product-specific guidances linked up to date with the appropriate general guidances.
Thank you, Dr. Lineberger. So, Dr. David Coppersmith, can you share some of your thoughts from a regulatory perspective?
Hi, this is David. Just one thing I wanted to add was, as my colleagues mentioned, that if there are differences between the general guidance and the PSGs, those are intentional differences. But if an applicant was to come across a difference that they did not think was intentional, they could utilize the controlled correspondence pathway to seek clarification from the agency on that difference. Thank you.
Thank you so much. With that, we are moving to our second question. So this question actually comes from our docket comment. Current FDA guidance recommends both fasting and fed BE for immediate release oral drugs. This is not aligned with other regulatory agencies. What is your perspective on this comment received in the docket? So with that, I want to come to Dr. Lei Zhang first.
Thank you, Lucy, for this question and comment. And FDA do acknowledge this comment, and we are aware that FDA's current recommendations on BE studies with regard to food is different from some of the other agencies. But because many products will be taken with meals, so currently FDA is recommending both fasting and fed BE studies pretty much for all immediate release oral products, with a few exceptions as explained in the presentation and also in the guidance, because of the concerns that the impact of food could be formulation dependent, and also we do not really recommend require the generic to be the same formulation as the innovator. So there's a potential risk that the fasting BE study conducted to compare the generic and innovative product, if they're BE under the fasting condition, may not be always translatable or predict whether the two products will be remain to be bioequivalent under the fed conditions. That's why we make these recommendations due to lack of a clear criteria we can use to predefine which product is at higher risk than the other products. However, FDA as well as other scientific community are having active research or existing research to understand the interaction of the food and the formulations from a mechanistic understanding point. This can help us determine whether we can utilize other tools such as some in vitro testing or in silico modeling approach can help us assess such risk and manage such risk. and help us determine the proper BE testing methods. For example, maybe there could be some alternative approach we can use in lieu of doing an in vivo BE studies. Currently, we also aware that recommendations related to fasting versus fat BE studies are under discussion as part of the harmonization efforts under the International Council for Harmonization, or ICH. They have a M13 expert working group specifically working on developing a harmonized BE guideline with a focus on the immediate release oral dosage form drugs. So at this point, we defer to the ICH process for any updated recommendations in this regard. Now I'm going to turn over to Niloufar, who can add some more perspective from a review perspective.
Thank you, Lei. Yeah, so I agree with all what Lei mentioned here. The recommendations in this guidance don't align with the recommendations for many other agencies. But also at this time, our expectation is that the recommendations in the current guidance would be followed. Whatever we have in our PK guidance as well as the PSG guidance for FET studies under the IR. And when M13 is finalized, we will adopt whatever gets described in the M13. And also, like from the review perspective, of course, we know that the applicants always have the option of an alternate approach for if they don't want to do the FET study. So if you can make a strong case for your alternate approach and it meets the statutory and regulatory requirements, that option is always available to you. Also, we also in our guidances, including our PSGs, we clearly mention that the recommendations are our current thinking and they are not binding. And lastly, the point I would make is that we recognize that the science of equivalence is evolving, and we try to ensure to the extent possible that the scientific the scientific standards will keep pace with it. And based on the knowledge and experience that we gain in a certain area, our thinking and recommendations will evolve. So I hope that answers the question. Thank you.
Thank you, Lei and Niloufar, for sharing your inputs. Can we move to the next question, please? So the question number 3 is, the revised guidance recommends applicants to include similar proportions of males and females in the study or provide a justification supporting the use of a single-sex population. What are FDA's considerations that lead to this revision to recommend including the justifications for not including subjects of both sex in a BE study? So Dr. Liang Zhao, can I turn to you first for this question?
Sure, thank you, Lucy. It's a very good question. FDA encourages the applicant to be inclusive and include both males and females in the BE studies if products are intended for use in both sexes. In general, as presented earlier, sex-related PK difference is assumed not to be affected BE conclusion. However, if combined with the formulation differences and related effects on absorption, sex-related difference in PK may potentially affect BE conclusion. Formulation physiological interactions may be different between test and reference product. Relevant physiological factors including pH, fluid volume in GI, gut transit time, pre-systemic metabolism, etc., may be different across age groups, across sex groups. After you expect the justification should explain that a drug or formulation specific factors are not likely affect BE conclusion when the applicant's BE study was not inclusive as recommended by the guidance. Credible sources such as NDA reviews available to the public the scientific publications, as well as in vitro studies, formulation, including formulation characterization, clinically relevant dissolution studies, and modeling simulation. For example, population PK to include sex and age groups as the covariate for PK exposure, mechanistic modeling such as PPK to test impact of study population choices, etc. All these approaches can be used for justification. Thank you.
Dr. Zhao, thank you for sharing the scientific thinking behind. So I want to turn it over to Dr. Diana Vivin to share some of your inputs from the ENDA review perspective.
Hi, thank you, Dr. Zhao, for that comprehensive look at the reasoning behind this change. I just want to reiterate from previously presented by in the presentation that we're really not making a major change here. We've always recommended applicants to include similar proportions of males and females in the study. Really, the change is just the opportunity to provide a justification to support the use of a single-sex population if that's chosen. So we want to encourage people to include in their study design similar proportions of males and females. We recognize that it may not be an issue in certain cases, so you can provide a justification of what you think is scientifically appropriate for use, as Dr. Zhao had mentioned. Thank you.
Thank you, Diana. With that, we hope that the response is clear to you all. We are going to move to question number 4. The revised guidance recommends applicants to include as many subjects as possible at or above age 60, or provide justification if no subject at or above age 60 is included in the BE study, if the drug product is predominantly intended for use in the elderly. What are FDA's considerations that lead to this revision to recommend including the justifications for not including older adult subjects in a BE study? So I want to start from you, Dr. Bini.
Thank you, Lucy. I think this question actually shares the similar consideration with the previous question related to our recommendation with the subject sex distribution. I can add a little bit on my end. I think ideally, a clinical study would fulfill its best purpose if the study is conducted on the intended population for its treatment. And what we are recommending here is intended to encourage the applicant to conduct their BE study on their intended use population. And such practice would allow us to obtain the data that shows the formulation differences on that group of population. And in addition, if there are any differences of the test and reference product that could be captured by this, in this case, elderly population, but less sensitively detected in the younger population, this optimized study design would give us a clearer picture. So we encourage the applicant to incorporate this consideration when they think about their BE study design and recruit the subject for the BE study. Thank you.
Thank you, Bin. So Dr. Robert Leinberger, can we also hear some of your thoughts in this question?
Sure. I mean, I think, echoing what you've heard before, again, in all of these questions about justification, right, there's nothing new in terms of the recommendation. This has been in FDA's guidance for many years. Here, what the change is really is asking for a justification if you really completely ignore this, right? If you look at this particular panel, if no subjects, right, you didn't find anyone included in the study, then you should think about that. And I think this is important also. If you think about why we're asking for this, right, is that you all, as speaking to the audience of people who are developing generic products, right? You know the population of people that are going to use the products that you produce, right? And you, of course, your organizations, have the goal of producing high quality products that are going to be appropriate for use in the populations for which they're intended. And that's a core part of the product development process. The way I think about it, there's really two scientific aspects to it. There's an aspect of clinical pharmacology and an aspect of the pharmaceutical or formulation science. And you have to put those together to do that. And that's what we're looking for in the justification. This is an important part of pharmaceutical development. From the clinical pharmacology side, what are the differences between the population that I included in my BE study and the population which will use the product? And you think about the interface of that what are the differences in formulation between my product and the brand product? And is there any interface between those two considerations? That's sort of the fundamental scientific question here. But that's something that every company that's developing a generic product, right, has to be considering now. Right? This is nothing new, so you should be able to put this piece together very clearly because it's sort of fundamental to the core way you have to think about this. And again, there's nothing in this, we, like everyone else at FDA, believe that bioequivalence conclusions, especially from crossover studies, are highly transportable, right? But you want to make sure that there's not any sort of interaction of the formulation and the subjects, thinking about both the formulation science and the clinical pharmacology that would render that assumption not correct in a particular case. And that's why we were asking people, and this sort of says, if you really end up without any of those subjects in your studies, you need to think through it. We're looking for the justification for that difference in terms of thinking through the clinical pharmacology and the formulation differences to identify that there's no additional risk for the patients who will use your product. And this is something that's in, again, the best interest of any organization that's developing the generic products.
Thank you, Dr. Lineberger. Thank you so much for sharing your perspective in terms of the interplay between the clinical pharmacology differences among different populations and the formulation differences. That's an excellent point. Thank you. With that, we're moving to our last question, which is question number 5. The revised guidance recommends applicants to justify that the BE study results obtained from adult subjects are relevant to the pediatric population if the drug product is predominantly intended for use in pediatric patients younger than 6 years. What are FDA's considerations that lead to this revision to recommend including the justifications for results obtained from adult subjects are relevant to the pediatric population for certain products? Dr. Yang Zhao, can we come to you first?
Yeah, sure. Glad to address the question. In general, a BE assessment in adults between two products can be used to support a BE assessment in pediatrics. If the product is intended for use in pediatrics patients younger than 6 years, the applicant should justify that the BE study results obtained from adult subjects are relevant to the pediatric population. Under 6 years old, physiological factors, again, can be more different from adults than older children, including GI motility, volume, transit time, that will impact potentially age-related absorption. Also, enzyme expression levels could be different, which will impact age-related metabolism, including pre-systemic metabolism, which can potentially impact BE results. Clinically relevant dissolution testing or other relevant in vitro approaches and mechanistic modeling simulation, PBK, may be included as the justifications. Modeling simulation can play an important role here for the justification and evaluate the impact of different enzyme on toluene, ADME properties in pediatrics on BE. So we have in the past have some publications in this area. If the audience have interest, we can supply the references in a different venue or after this meeting. Thank you very much.
Thank you, Dr. Zhao. Could I come to you, Dr. Larufa Temple, to share some of your inputs from the ANDA review perspective?
Sure, Lucy. Thank you. So from the review perspective, right, as part of the justification, our guidance here also is mentioning to consider the safe use of the excipients in the drug products meant for the pediatrics. And within the Office of Generic Drugs, assessing safety of the excipients is already a long-established practice. So again, this is not anything new that we have introduced. We kind of, for all applications, we assess the short-term and the long-term safety of the excipients for the intended population. So we assess the safety in the context of use, of how this drug is intended to be used. What population is it meant for? Is it adults or pediatrics, or is it a special population? Is it for acute use versus a chronic use? And we know, like pediatrics, they are not like young adults, and excipients which are commonly used in the adults may not be safe for the pediatrics. The toxicological risks and the safety concerns are greater for the pediatrics. And as Dr. Zhao mentioned, that's because their GI physiology and their anatomy are different from the adults. We have other physiological factors which are different. For example, the metabolic enzymes. In pediatrics less than 6 years, they are not as matured. And then in that case, there is a risk of accumulation of the toxic molecules in absence of certain enzymes, which may not be the case for adults. So there is, and also one other point is, this is the reason why we also don't lump the children of all ages into one group for this assessment. We know that changes in the metabolic processes and other changes, they continue through their rapid growth periods, from moving from the neonates to the infants to pediatrics less than 6, pediatrics greater than 6, etc. So I hope you can appreciate why this recommendation was added to the general guidance and why it's important. Thank you.
Thank you, Dr. Temple. I want to take this opportunity to thank our panel members for spending your time with us, addressing the questions. I thank you so much. And with that, we're going to conclude our panel discussion session, and I will turn it back to Ray to start our Q&A session.
Thank you, Dr. Feng, and we want to thank all of our panelists for the great discussion and for all the great presentations for our presenters. We'll now transition into the Q&A session. And just as a reminder, if you haven't had a chance to enter your questions relevant to the presentation into the Q&A chat pod, please do so now. We'll answer as many questions as time allows. Looks like some questions are coming in right now. The first two questions will be directed to Dr. Bing Li. And here's the first question for Dr. Li. For various BE approaches stated in the guidance, such as lines 58 to 63, related to 21 CFR 320.24, can you clarify whether these various studies, such as comparative PK, comparative PD, comparative clinical endpoints BE studies, and in vitro studies, have equal priority instead of descending order of preference in the previous guidance?
Thank you for the questions. I think the descending order of the preference of the BE approach listed in the CFR 320.24, which is the Invivo PK, Invivo PD, Invivo clinical endpoint study, the in vitro comparison, and other approaches deemed appropriate by FDA for BE assessment, applies mainly to the systemic acting drugs. We actually take a comprehensive scientific approach to handle the bioequivalence on the locally acting drugs or some complex drug products. One example is, the locally acting nasal inhalation drug product, where a weight of evidence approach, which encompasses in vitro comparison, Nvivo PK study, Nvivo clinical endpoint study, are recommended. They are all weighing equally into the overall bioequivalence conclusion of the bioequivalence of a drug product. Thank you.
Thank you, Dr. Lee. And then we have another question for you before we transition to our next panelist, which will be Dr. Robert Leinberger. And here's the question for Dr. Lee. Are there any specific recommendations if a sponsor decides to use authorized generic as RLD due to business or formulation comparability?
Authorized generics are identical to the brand product, and again, they can be used in the bioequivalence studies as the reference standard. So it's completely appropriate to use the authorized generics. Those are produced and approved under the NDA of the RLD. They're exactly the same product. I think FDA still gets a lot of these questions and control correspondence asking if this is okay. It's a long-standing question that has a standard answer.
Thank you for responding to that question, and it looks like we may have another question for Dr. Leinberger. Let me just 1 second while there we go. My document repopulates, and here is the next question for Dr. Leinberger. What is FDA's perspective on limiting to certain population types, such as Asian only, Caucasian only, for BE studies, and typically single-center studies conducted at one geographical site, if conducted outside of North America, may not have a diverse ethnic subject pool?
Again, so in the revisions to this guidance, that factor is not mentioned in this guidance revision here. So that's not something that we're talking about in this revision. Again, some of the same principles may apply. All right, especially you go back into what I say about if the clinical pharmacology says there are differences in these populations that are relevant to the drug, and the formulation differences interact with those in some way, then certainly there may be something that you might want to think about or consider in what sense the information is needed. But there's not a specific recommendation in this guidance about differences in ethnic groups or the regions in which you may do the bioequivalence study in this case. But again, there may be situations for a unique product where the considerations may apply. But again, we don't have a general recommendation around that or are looking for anything specific at this time.
Thank you, Dr. Leinberger, for responding to that question. We'll be moving on to our next panelist right now. The next panelist will be Dr. Diana Vivian. And here is the first question for Dr. Vivian. Can you further elaborate about the type of cases where an in vivo BE study may be needed to compare a proposed modified release generic product to the RLD for alcohol effect when there is no PSG?
Thank you. Sure, I can address that. So first I would say if there is no PSG for this product, I'm not aware that we recommend an in vivo BE study administered with alcohol as a first line of thinking for any PSG. If you're concerned that there may be effects of alcohol on a drug product, you can certainly submit a controlled correspondence or some kind of communication to the agency. So this would be in cases where you suspect there may be a safety or efficacy concern for a modified release formulation due to alcohol, in that case, we would typically recommend an in vitro assessment first, and only if there's an issue with the results of the in vitro assessment. So, for example, if there's much more release of the test product than the reference product, and we think that it could be a clinically significant impact on safety, for example, then we may, in rare cases, ask for an in vivo BE study to be conducted. If we do ask for that, we would ask for the protocol to be submitted before the conduct of the study to ensure that we are okay with the study design, or if we have any recommendations to ensure study subject safety during that study. So I would never go straight to an in vivo BE study with alcohol if you think it may be a concern. Thank you.
Thank you, Dr. Vivian, for responding to that question. We have one more question for you before we move to our next panelist. And here's the question for Dr. Vivian. Is it okay not to bioanalyze the subjects who are withdrawn due to AE or other reasons and do not complete all periods of study, particularly in a full replica?
Thanks. So I think this is referring to the change in the current guidance recommending that outliers not be excluded. So in this case, if you're pre-specifying that you will not bioanalyze a subject who's withdrawn due to something that's justifiable, like a adverse event, for example, I know in our current guidance, we recommend to exclude data if a subject takes an IR product and then vomits before 2 times the median Tmax. That may be an example where you put in your protocol, if that happens, then you're not going to bioanalyze that subject. And that may be acceptable, and we wouldn't consider that a statistical outlier. When you say particularly in a fully replicate design, we have had cases that we've seen of people excluding or not bioanalyzing subjects because of AEs or other concerns that are pre-specified and justifiable. I would also recommend that if a subject does complete at least 2 periods of the reference product, that can be really useful information as far as accurately representing the reference product variability. So I would encourage you to keep those subjects and analyze those, even if they may not be used for a TR comparison. For a fully replicated design, they could be used to estimate the reference product variability. Thank you.
Thank you for responding to that question. We're moving on to our next panelist. And next panelist we're bringing in is Dr. Nilofer Tampel. And here's the question. It has several parts for Dr. Tampel. There are several questions related to BE for additional strengths of MR products. And here's the first part. Why do we need to meet the condition the reference product demonstrates dosage form equivalence among different strengths and demonstrates similar dissolution performance across different strengths? Second part of the question, please clarify what is meant by non-proportional in the bullet. Strengths can be either proportional or not proportional in quantity. And finally, could you also clarify the purpose for generating dissolution data on all strengths? Dr. Tampel, we cannot hear you if you were speaking. What we're going to do is move on to our next participant on the panel.
Rick, can you not still hear me?
We can hear you, Dr. Tampel. We'll let you go right ahead.
Okay, thank you. Right, so I was saying like dosage, I'll start with the dose strength equivalence question. That was the first one. So this dose strength equivalence, right, it informs us if the RLD exhibits consistent bioavailability between the strengths. So for example, we have 3 strengths like 10, 25, 50, then the dosage strength equivalence will test 10 of 5, 2 of 25, and 1 of 50 in vivo to show that there is consistency of the dose, right? And then sometimes the RLD may not be formulated to be compositionally proportional, in which case we need to have this information on the dose strength equivalence or dose strength proportionality. And in other cases, it could be that the strength RLD is compositionally proportional, but say the in vitro release rates are different between the strengths. We need to assess that data and see like how this different dose strengths will behave in vivo. So again, this dose strength equivalence study would be kind of recommended. So we have all this information from the RLD, which then like during the PSG development, we assess this information to determine if the alternate approach for determining bioequivalence would be applicable to all strengths, meaning if we cannot ask for an in vivo study. And so that's how we assess this. And at most of the times, the PSGs would be available. So then maybe you may not have to really go dig for this information. So that is what I would say for the dosage strength. Okay, so the next one was proportionality, correct? Okay, so for the proportionality, I think the guidance describes how we assess the proportionality. So very briefly, if I can say, it talks about there are 2-3 bullets there in the guidance where the active and the inactive are in exact proportion, or we have the same inactives within the formulation and the change in any one strength is achieved by altering 1 or 2 excipients and the amount of the active. And this kind of is applicable where the total weight remains sort of similar, like and similar would mean within 10%. So that may be simple to follow for the IR. For MR products, it's a little more challenging since they can be formulated in different ways. So they will have also different release mechanisms. So you can, if it's formulated, say, as a tablet with layered formulation, so which you have a core in the center, then you have an ER coating, then you have a DR coating. Or you can have a matrix formulated with an ER coat, or you can have an osmotic pump. So different types of formulations. So in some cases, like we can't be assessing the proportionality of this type of different formulations without understanding the mechanism of release and what's critical in those formulations, right? So in some cases, we can assess the drug to excipient ratio for the proportionality, and that may be appropriate. In other cases, the proportionality of the rate controlling and non-rate controlling, they may be assessed in terms of the amount of excipient to the total weight of the rate controlling and non-rate controlling excipients in the formulation. So, and for this, we would generally draw from the principles of the SUPAC guidance. And although SUPAC is really meant for post-approval changes, but the principles of those is where we could draw on those for determining proportionality. And that's where, if we talk about proportional, then if we don't meet all these criteria, then that would be non-proportional. And the next question was on dissolution, right? Okay, so for the dissolution, we assess the test formulation design and the dissolution data at multiple pH. And as part of the totality of evidence, which was kind of explained during the presentation, we will consider if the in vitro dissolution profiles of the RLD and the test at multiple pH conditions are, whether they're similar or they're significantly different. And if they are significantly different profiles, it may suggest for the test product that there could be a risk for not having the same efficacy, safety as the RLD. And so that's why we consider the totality of evidence, and for us to do that, we need all the data. So we need the data for all the strengths of the test product as well as the reference at multiple pH. And just one last point, if I could make, that in the current guidance, rather than proportionality of the formulation composition as a dominant factor, emphasis is also on consistent drug release from in vitro dissolution testing. So each of these factors become important to assess whether the alternate approach can be applied, or do we need an in vivo study. Thank you.
Thank you, Dr. Temple, for responding to that three-part question. We're going to move on to our next panelist. The next panelist that we'll bring into the discussion is Dr. David Coppersmith. And here is the question for Dr. Coppersmith. Are applicants required to follow the recommendations in the guide?
Thank you for the question. So as my colleagues have mentioned, the guidance provides recommendations. They are not requirements unless they are specifically citing to a statutory or regulatory requirement. So in that case, the guidance would use the words, some mandatory language such as must or required. But when the guidance is using the word should, that's obviously referring to something that's suggested or recommended, but not required. But guidances do reflect FDA's current thinking on particular topics, so I do encourage applicants to consider the recommendations and suggestions included in the guidance, although they are not required. These applicants can propose and utilize alternative approaches if they meet applicable statutes and regulatory requirements, but they should be encouraged to review the recommendations and suggestions because they, as I mentioned, they do reflect FDA's current thinking. Thank you.
Thank you, Dr. Coppersmith, for responding to that question. And we have another question before we move on to the next panelist. And here's the question. If a drug was issued CGT exclusivity, is there a possibility for another drug to get full approval before completion of the CGT exclusivity?
So, thank you for the question. So CGT is referring to competitive generic therapy. In the 180-day CGT exclusivity period described under the Federal Food, Drug, and Cosmetic Act is triggered by the first commercial marketing of the CGT drug by the first approved applicant. And then after this exclusivity is triggered, the 180-day period would run without interruption. Once the first approved applicant has commenced commercial marketing, FDA is restricted from approving ANDAs for a drug that is the same as a CGT approved in the first approved applicant's ANDA. So FDA would not be restricted from approving other ANDAs covering a drug that is the same as the CGT prior to or after the approval of a first approved applicant's ANDA for the CGT, unless and until the first approved applicant has commenced commercial marketing and triggered that exclusivity period. And if you would like additional information on CGT exclusivity, I would refer you to the CGT guidance, which was issued, I believe, back in 2000, which has quite a bit of information in it. Thank you.
Thank you, Dr. Coppersmith, for responding to that question. We're going to move on to our next panelist. And the next panelist will be Dr. M.J. Kim. And here's the first question for Dr. Kim. As stated in the presentation, that if the label suggests that the product should be taken on an empty stomach means in fasting conditions. Still, product-specific guidance of this kind of product requires a BE study in both fasting and fed. Why is this?
Thank you for the question. So I want to kind of clarify regarding the need for both fat and fasted. When we develop PSGs, we look at the product, the labeling, the product labeling very carefully, and we try to assess whether a drug should be given under both fat and fasted or under empty stomach. In the past, we have noticed that some product labels may not be quite clear as to whether a drug should be given under empty stomach, and there may be some ambiguous or some unclear labeling instruction. So about 2 years ago, what we did was we looked at all those products and we kind of clean up the PSGs. So if you were to take a look at particular PSG, and if the PSG recommends only a fasting study, then you should consider that as recommending just one study under a fasting. And there may be some kind of confusion as to what about the fact that general guidance recommends both fasting and fat studies, but once again, it's for the immediate release products. We have a general recommendation, and then but then the labeling specifies, and you heard from Dr. Lei Zhang's presentation that we have given a very specific case where a fasting BE study may be recommended as an only study. And if in that case, it's when the labeling says that you should be given under empty stomach. And another scenario that you have seen from her presentation was in the event that there may be some serious adverse events that are anticipated under fat conditions. So we are very mindful about the safety aspect as well as what is recommended in the labeling. So when you do studies, please refer to the PSGs that you're looking at, and the PSGs with the fat and fasted that should reflect the latest thinking, and they're all updated, as I said about 2 years ago. Thank you.
Thank you, Dr. Kim, for responding to that question. We do have one more question before we move on to our next panelist for Dr. Kim. And here's a question. You sorely touched on in that answer you just gave, but we'll just go ahead and read this out for you. If the PSG recommends only a fast BE for an IR product, what is the need for a fed study which is included in the general guidance but not the PSG?
Yes, so this again touches upon the difference or so-called difference between the general BE guidance versus the PSGs. But if you really think about the general framework that we use for the general BE guidance, we do recommend fat and fasted studies, but then we look at a drug at a time, and we look at the drug properties as well as formulations, PK, clinical pharmacology aspects, and then we consider all that in addition to safety assessment of doing the BE studies, and with all this information kind of combined together, we come up with a specific recommendation as to whether a fasting or fat BE study should be done, and whether a study should be done in healthy or other target populations, and they're accurately, and we try to be accurate as possible, and they're reflected in the PSG. So please refer to our PSG website to look for our latest thinking and the recommendations.
Thank you for responding to those questions, Dr. Kim. We're going to move on to our next panelist, and we're going to bring in, for our next panelist will be Dr. Lei Zhang. Dr. Lei Zhang. And here is the question for Dr. Zhang. Why are BA studies not mentioned in the scope of an ANDA?
Thank you, Ray, for that question. It's very interesting observation. I would not say BA is not in the scope, because we all know bioavailability is the foundation for the BE. So we do need to collect the BA information on both the test and the reference or reference list drug in order to, this is the statutory requirement for ANDA applicants to demonstrate, the proposed generic product is bioequivalent to the reference list drug. In order to do that, they need to assess BA for both products. So I will not say it's out of scope. Maybe it's not specifically mentioned as the other guidance, but it is a foundation for the BE. Thank you.
Thank you, Dr. Zhang, for responding to that question. We're very fortunate. We've got a few more questions for you. And here's the next question. It's a little bit longer, so we'll start reading it right now. It's described that the guidance based on the BE principles that the rate and extent of absorption of the drug do not show a significant difference from the rate and extent of absorption of the listed drug when administered at the same molar dose of the therapeutic ingredient under similar experimental conditions in either a single dose or multiple doses. From this principle, there's no concentration on the dosage form. This means that FDA can allow one to perform the BE study between two products with a different dosage form, such as a suspension versus a tablet.
Yeah, thank you for the question. I think this question I need to clarify. Although people can conduct so-called BE studies among different dosage forms, just for the sole purpose for bridging or for some other purpose to check the relative BA, but for ANDA or 505J application, the statutory required the two products to be at the same dosage forms. So strictly for the ANDA application, we look for BE among the two product with the same dosage form, same route of administration, same labeling recommendation, and etc. So this is the just regulatory requirement. However, as I mentioned in my presentation as well, is that the BE concept can be used for many other purposes. For example, to show the relative BA of the two different dosage forms, or for other, the 505B2 or some other applications when they try to show the equivalence between the two products, the test versus the reference comparator. So that is, they use the same principle. So that's the clarification I want to make here. Thank you.
Thank you, Dr. Zhang, for responding to that question. We're going to move on and bring in our next panelist. Our next panelist is Dr. Leng Zhao. And here is the question for Dr. Zhao. How can we determine if the drug product is a highly variable drug and or narrow therapeutic index drug? Has the FDA published such a list that we can reference?
Yeah, the highly variable drug is defined as, you can find many publications actually, that a drug within subject variability means if you dose the same individual twice, the variation is if it's bigger than 30%, I think the exact number is 29.4 if I'm correct, then is considered the highly variable drug. Then the drugs eligible to use reference scaled average BE analysis for BE assessment. We propose that approach is mainly to reduce the number of subjects that's needed to pass a BE and make it more reasonable for industry to have a generic regulatory path. So for narrow therapeutic index drugs, the similar criteria has been applied after the reference scaled ABE analysis being introduced for highly variable drug. Narrow therapeutic index drug have the nature of with little change in exposure that could lead to efficacy and safety concerns of the drug product. With that, we need to tighten the BE criteria. We have classified a number of NTI drugs in the product-specific guidance. We may have future communication publication down the road to download to the community what are the current list of NTI drugs. We also have a working group within CDER that been set up to identify drugs with NTI potentials. So, currently you can mainly rely on the PSGs in terms of finding all the NTI drugs, but we are applying the reference scaled ABE approach plus the ratio test for the within subject variability. Hopefully I have addressed your question in a satisfactory manner. Thank you very much.
Thank you, Dr. Zhao, for responding to that question. We do have one more question for you before we move on to the next panelist. And here's the question. There is no gender effect on PK. Is it okay to consider single-sex population for BE studies?
Yeah, as presented earlier by Lei, Nilufer, and others, and also as I mentioned it, certainly is applicable to use single gender in the BE assessment. However, if the drug is intended for the full population rather than the single gender, then I think you need to provide justification that the BE established in the single sex group can be extrapolated to the whole population. Usually that's the case. However, the extent of justification, if it's obvious, if there's publication, there's no gender effect in PK or sex effect in the PK, then you can, rely on those already known facts or findings based on data to as the means of justification to support the BE assessment or BE extrapolation. Thank you.
Thank you for responding to that question. We just had one more question come in for you, Dr. Zhao, and we'd like to just squeeze this one in. And here's the next question for you, the final one. Can an outlier subjects in a replicate study design be considered for exclusion from the study?
No, the general answer is no, unless that's been pre-specified in the analysis in the protocol at the experimental error or protocol violation or things of the same sort. Otherwise, the outlier do carry valuable information potentially for the formulation. So FDA do not allow post hoc removal of outlier data. Thank you very much.
Thank you, Dr. Zhao, for responding to that question. We're going to move back up to the beginning of our panel and bring back in Dr. Bing Li. And here's the first question for Dr. Li. Are there any specific recommendations if a sponsor decides to use an authorized generic as an RLD due to business or formulation comparability?
I think Rob has briefly touched upon this question. In general, I think the answer is yes, provided that the applicant can demonstrate that the RLD is not available. This question here does not provide the reason, the detail of the reason, just simply says business or formulation comparability reasons. Without the detail, especially formulation comparability reason may not be regarded as a warranted reason for requesting an authorized generic be used in the BE study. Thank you.
Thank you, Dr. Lee, for responding to that question. We do have one more question for you, and here's the question. Does the understanding on the applicability of PK study results in adults also apply to children over 4 years of age, in particular for inhalation products?
Yeah, thank you for this question. In general, as we discussed in the panel discussion, that a BE assessment in one population can be used to support the BE assessment in another population. This is because, as we discussed, that for a bioequivalence study, since the purpose of the study is to compare two different formulations, and it is generally believed that the physical process of the drug absorption after it released from the formulation are usually the same across the different populations. So in general, we believe the BE conducted in one population could also transfer to another population. In this current guidance, as you learned from the earlier discussion on all the rationales and reasonings, that we made some additional recommendations related to pediatric use drug product. That is, if the drug is predominantly intended for the use of pediatric younger than 6-year-old, then the applicant is recommended to actually think about two additional considerations. One is to justify that the BE study results obtained from the adult subject are relevant to the pediatric population. That's related to the bioequivalence study outcome. The other one is a safety consideration related to the inactive ingredient used in the formulation, which is to justify that the inactive ingredients used in the proposed product are appropriate for the pediatric use population as well. Thank you.
Thank you, Dr. Lee, for responding to that question. We're going to move on to our next panelist, and we do have another question for Dr. David Coppersmith. And here is the question. If the RLD has been discontinued from marketing, can the applicant use a reference standard instead?
So when in vivo bioequivalence testing is required or an applicant otherwise seeks to support a demonstration of BE with in vivo data, such testing under the regulations must be conducted with the drug product that FDA has selected as the reference standard. Where the RLD is marketed, ordinarily it is also the drug product selected by FDA as the reference standard. Where the RLD, though, has been discontinued from marketing for other than safety or effectiveness reasons, FDA may select a different listed drug as the reference standard, but in either event, the reference standard would be the drug product that would be used for the bioequivalence testing. Thank you.
Thank you for responding to that question. We have just a few more minutes remaining in the webinar, and we would like to thank all of our panelists and on the Q&A session for answering numerous questions that came in. It's my pleasure to turn the webinar back over to Dr. Lionberger to provide some closing thoughts.
Thank you very much, Ray. So I appreciate all of the work that went into the staff from our SBIA group, as well as the FDA organizers and speakers, especially Lei and Nilofer, and all of the panel members who put this opportunity together to have an opportunity for you in the industry to ask questions about our new bioequivalence guidance and our bioequivalence program in general. we were not certainly able to answer everyone's questions, but we did get to see all of your questions, and we'll certainly take them into account as we prepare different opportunities to communicate to you about the generic drug program and bioequivalence. A lot of the questions that we didn't answer were a little bit more general than the new changes in this guidance. We saw a lot of questions about how our bioequivalence system works or how the generic drug program works that are a little bit beyond this question. So we'll look for other opportunities and at other SBI events, such as the one that's coming up in April, to address those more general questions about how our program works. But really appreciate all of the people who worked hard to prepare the slides for this and to give this. We hope to do this type of event for every new scientific guidance that we put out to really provide a deeper thought and discussion around why we're doing different things in the guidance. Hopefully that makes it clearer to you, the attendees, the people in the pharmaceutical, people in the generic drug industry who are using this guidance, the scientists and product developers and regulatory affairs experts in your organization, that you can understand, not just what's written in the guidance, but help you understand on a deeper level what the thinking is behind this and how it fits together. And so that's the reason for doing this. And I hope that you've gotten that here and you've seen, especially in some of the new things here, the really, important interplay between thinking about, as you're developing the drugs, thinking about the clinical pharmacology and the formulation science of your products. Those two are the core pieces of the bioequivalence science that drives the generic drug program. And it's really important for you to begin, you in the industry to begin to develop and build the understanding that you have in this area in order to develop products effectively, to use the regulatory program for ANDAs effectively, is to understand that. And I think so this will be an ongoing dialogue and discussion. We look forward to interacting with you in various ways and helping you to navigate through the development of generic products in an efficient way and helping to grow your organization's capabilities to be able to do this in an efficient way and do things that also not just help you meet the FDA regulatory requirements, but also help you move toward high quality products for your customers, for the patients, and that's the goal of all of this enterprise that we're engaged in together. So with that, I want to pass back to Ray for some closing administrative details, and really again thank Ray and the other SBIA staff members for another excellent webinar with really smooth logistics and just fantastic to work with them and the great job that they do in helping us communicate things to the stakeholders for the generic drug program.